Good morning everyone. I am Muralidharan. I have given 4 UPSC mains. I have been selected through UPSC for the post of Enforcement Officer in Employees Provident Fund Organization. Today we are going to discuss Biotechnology syllabus for UPSC preliminary examination. It comes under the Science and Technology part. To understand the significance of biotechnology in preliminary examination, if you go through the past 10 years paper, you will come to know that at least one question is directly from biotechnology. In addition to that, your GS3 paper under mains, there is a syllabus for biotechnology. So there in mains, in GS3 mains, you can expect 1 10 mark or 15 mark for biotechnology. Mostly it includes the scientists, the biotechnology scientists in India or the recent advances. Once a question was asked, how biotechnology can be used to improve the income of farmers. So such questions are asked. Okay. So today we will go into the biotechnology part. Before going into it, we will see the previous year questions, for the previous 10 years questions. So you will have an idea what UPSC is expecting from us, what UPSC wants to know in biotechnology. Okay. So let us go through some of the previous year questions. So I have categorized the questions into 3 types. Okay. For example, UPSC is asking you what are the recent advances in biotechnology. It is directly mentioning those advanced technologies and asking what is the use. That is one type of question. The other type is UPSC is asking, it is mentioning technology and what are its applications. So you should know the application part of it. And the third thing is sometimes, unfortunately, UPSC is asking the core concepts. You should know the core of the biotechnology. Only then you can answer such questions. Okay, so we will look into it. So please see this question. This question, if I'm right, it's in 2019. Pro-nuclear transfer. So here they are directly asking what this technology is used for. Then next question about Cas9 protein. So what is Cas9 protein, which is often mentioned in news. So they are directly asking what it is meant for. Then see the next question, application part. So BT brinjal, what is the use of BT brinjal? Then the next question, transgenic plants. What are the uses of transgenic plants? What are the applications of transgenic plants? So this question was asked in 2012. Then in 2013, mycorrhizal biotechnology. What is the application of mycorrhizal biotechnology? That is how fungal biotechnology, what is its use? So that they have asked. Then, what are the applications of RNAi technology? Okay. Then, here, now we have seen what a technique based question, application based question. Here they are asking concept based question. Okay. So, see for example, this question asked in 2019. See the first statement. Functional chromosome can be created by joining segments of DNA taken from different species. Can you understand? They are telling you can take DNA from different species, say bacteria, plant, animal. You can join them and you can make one functional chromosome. See the statement 2. Pieces of artificial DNA can be created in laboratories. They are telling you can synthesize a DNA in a laboratory. See, to understand such, so you should know the core of biotechnology. Only then you can understand whether such statements are right or not. See, statement 3. A piece of DNA taken from an animal cell can be made to replicate outside a living cell. See, such statements mandates you to know some core concepts of biotechnology, and I am sure I will cover this in our class. Okay, see, and here genetic engineering allows transfer of genes. So here they have given comments. So genetic engineering, so you all would have read about transgenic plants, BT cotton, everything. Here they are asking whether genetic engineering is possible between whether you can transfer a gene from a bacteria to a plant, from a plant to an animal, from an animal to a microbe. So they have given various combinations and they are asking whether it is possible. So again here you should know that concept. Only then you can answer this question. Then this question asked in prelims 2018. See here they have asked about GM mustard. See here they are asking to what pest it is, whether it is against broad variety of pest or it is against a narrow case. And here they are also asking which institute has developed. GM mustard is developed by Indian Agriculture Research Institute and Punjab Agriculture University. So they are also asking which institute is developing. So we have to read, we have to cover our biotechnology in these aspects. So what are the latest technologies? What are the various applications? And 3, some of the concepts. Of course, we cannot cover all in-depth concepts. We are not going to do any research, but some basic concepts which are in use, we should understand. Okay. Okay, I'm sure. And all the answers for all these questions we'll see as we cover the syllabus. We'll discuss these questions and their answers. Okay, fine. To enter into this, before entering into this, you should know what is cell. Before going into advanced concept of biotechnology, we should know the some basic biological concept with which only we can build this subject. Okay, because sometimes UPSC is also asking a basic biology questions. Once, some few years back, there was a preliminary question. I think so. It is 2012 or 13 paper. They have asked about virus. They have given 3 statements about virus and asked whether it is correct or not. The statements are: viruses lack any enzymes to multiply. Viruses can be cultured in any synthetic medium. So these questions makes you to know some biology part. And this year, this prelims 2020, they have asked a comparison between plant cell and animal cell. They have given 3 statements about a plant cell and animal cell and asked us to choose which is correct. So here, before going to biotechnology, you should know some basics of biology which will help you. Okay. So, yes, to begin with the basics of biology, let us start with a cell. Okay, so we are all made up of cell, right? So we all know cell. We are all just like atom for we are all made up of cells. Cell. So the cells join to form tissues. Then the tissues join to form an organ system. Then the organ system in turn forms an organ. So it's a group of cells form tissues. Then tissues form a system like a respiratory system, digestive system, circulatory system. This system together joins and forms a whole organism. Okay, so that I have put. So cells combine to form tissues, organs, organ systems and organisms. Here our interest is a cell. Okay, here what I have shown is an animal cell. Okay, so we'll discuss that question asked in 2020 prelims also. They have given so how an animal cell is different from plant cell. That was the question. The major thing is animal cell has only a plasma membrane, whereas a plant cell, in addition to this, it has a cell wall. That was the statement there. Okay, so a plant cell will have a cell wall, then a plasma membrane, but cell wall is lacking in animal cell. Point 1. Then point 2 is vacuoles. Okay, vacuoles are absent or small in animal cell, whereas it is large in plant cell. Okay, then cell organelle called chloroplast, that is present only in plant cell and not in animal cell. Okay, so this will give you a fair idea of difference between plant cell and animal cell. Then coming to an animal cell, the structure of cell. So the cell consists of a plasma membrane, and inside it you have a cytoplasm and nucleus. Okay, a plasma membrane, cytoplasm and nucleus. The cytoplasm contains various cell organelles like mitochondria, endoplasmic reticulum, ribosomes. This you would have read in your school, 6th, 7th, you would have read this. Now inside this you have a nucleus. Okay, and in biotechnology our concern is this nucleus. This nucleus part is what we are going to deal with. Okay, fine. Yes, one thing I have noticed, especially among engineering students, they confuse between what is nucleus, what is chromosome, what is gene, what is DNA. Okay, there are many students who consider these are all different. A gene is different, DNA is different, chromosome is different. So that is not the case here. You should understand. I'll explain this with you. I'll explain this with a diagram. Okay, see, this is a cell. Okay, so this is a cell. Inside the cell you have a nucleus. Okay, this nucleus carries a genetic material, that is DNA. How in normal situation this is coiled. Okay, this genetic material is coiled and we call it as chromatin reticulum. Okay, and we call it as chromatin reticulum. This chromatin reticulum, then when cell undergoes replication, okay, here I will draw this forms a structure called chromosome. So this is your chromosome. Okay, now if you unwind this chromosome, is okay, so it will be like a dumbbell. Okay, so it will be like a dumbbell. Okay, that's we have this 23 pairs of chromosomes. So this condensed coiled material will arrange in a dumbbell shape called as chromosomes. Okay, this is chromosome. Now I am taking, if I unfold this, how? So this particular region I am taking, I am unfolding. How it will look like? It will look like this, like two snakes intertwined. Okay, like two ropes intertwined. It will be like this. So this is called as DNA. Okay, this is called as DNA. Then what is gene? In this chromosome, a particular part of this chromosome will code for a protein. It will code for a protein, it will code for an enzyme. So such region is called as gene. A part of chromosome that codes for a protein is called as gene. Okay, so here chromatin reticulum, then chromosome, then you have within the chromosome the functional part is called as gene, then you have, then this is all made up of DNA. Okay, so here please look into the slide, it will help you to understand. See, this is your cell having nucleus. This is the chromosome, and here the part of chromosome which codes for any protein is called as gene. If you unwind the gene, see for example this is the gene. If you unwind and see, it will be a double helical structure. It will be like a 2-rope coiled. Okay, that double helical structure is called as gene. Okay, sorry, DNA. Here